Good to see you again, Amos. So I would just like to introduce a friend and colleague. So Amos is speaking from University of Ibadan. I had the privilege to actually visit his place and meet Amos and his beautiful family. And we've kept touch since then. It's really been a joy to get to know Amos. He's really a brilliant guy and also a very a good person. In his talk, Drosophila melanogaster, emerging model system in toxicology. Amos, I'm looking forward.
Thank you very much. It's a great opportunity for me to be here. I'm grateful to DrosAfrica as well as Professor Sagan for this opportunity. My name is Amos Abolaji from University of Ibadan, Nigeria. I will be speaking on the Drosophila melanogaster as an emerging model system in toxicology. By way of introduction, I will first of all like to talk generally about toxicology for those who are not in this discipline, and I will quickly go through a brief introduction with respect to toxicology, after which I will go to toxicology, which is the subject of today. Societal toxicology defines toxicology as the study of the adverse effects of chemicals, physical agents, or even biological agents on living organisms and ecosystems. And this toxicology is not only limited to this, it also covers the prevention and amelioration of such adverse effects. It's really a multidisciplinary science that requires different professionals from different fields coming together to do different aspects of toxicology. These major areas are covered in toxicology. The first is developing mechanism of toxicity. It is not enough to say a particular toxicant or drug is toxic. It's also important to know the mechanism of toxicity. Then toxicology also involves ensuring safety of chemical products. Without carrying out toxicological studies, it will be difficult to really know the safety of some of the drugs and the medicine that we use. It's also important to ensure safety of so there are different toxicological studies that are they carry out on what. Lastly, evaluating risk linked with exposure to toxicants, and then to be able to determine the possible therapeutic approach to use. There are several branches of toxicology, as you can see in this particular slide. The ones that I highlighted are the aspects that are of interest to me. And if you look at all the different branches, you will realize that they are also linked, interrelated. There are several chemicals, biological agents, and also several other contaminants in the environment. At present, we have more than 80,000 commercial chemicals, and on a yearly basis, about 2,000 chemicals are being introduced, and we have limited data. The data available are not enough to be able to determine the mechanism of use of some of these chemicals. Air pollutants is in different places. Some of the air pollutants that we have are carbon monoxide, hydrocarbon, arsenic. It's important to take note of the fact that it's not only air that can be polluted, water also can be polluted, soil can be polluted as well. So pollutants are just any substance or matter that is in the wrong place in the environment. And a very important question that we can ask, is it possible to return the soil and water to them that we actually have no other answer. By way of a case study, we read, and also it was reported, the case of lead poisoning in the northern part of Nigeria, referred to as Zamfara State. In this particular region, as a result of mining process, soil and water were contaminated by lead, and as you can see, a child is seen in this slide that is in this environment, which should not be. And in that particular period between March to June 2010, about 163 adults as well as 111 children died due to exposure to this lead in the soil and in the water. Apart from lead, we also remember the contamination with mercury that happened at the Minamata Bay in Japan, where plastic and chemical plants were discharged, and then mercury present were converted to methylmercury, taken up by the fish, and fish get consumed by man, and then as a result of this, people die. Contamination by lead. It's difficult to toxicants or metal in the environment. If you look at mixtures of toxicants in the environment, for example, from waste, from industry, as well as pesticides, fertilizers, all these can get into the water sources and then even into the home. Eventually, man can be exposed to it. As a matter of fact, there is a branch of toxicology that is referred to as toxicology of mixture, that in reality, we are exposed to myriads of these toxicants. And the very sad thing is the some of these toxicants, it can affect the damage that can eventually have adverse effects when the child is born. Environmental toxicants have several fates after either through inhalation or ingestion. It can get into the bloodstream, get metabolized. The metabolite itself can be more toxic than the parent compound and cause toxicity, or it can also be stored in certain tissue and bioaccumulate. But the whole process that the body will use is just to find a way of excreting the toxicants from the system. We have several actions of this toxicants on the system. For example, if a woman is exposed to drugs, cigarettes, or lead in the during pregnancy, it can pass through the placenta and affect the child. It can also, some of them can also cause carcinogenesis, organ specific. For example, we have reproductive toxicants, and we also have tools in toxicology, even white blood cell as well as Drosophila that we are considering. It's important to highlight, based on this particular conference, on this session, toxicology, which is just the use of the truly an emerging field. New studies are being carried out, and the new method of carrying out different assays are being, different markers are being developed. Let me just use just a minute to let you know that I did not start my career with Drosophila. I actually started with rats, like most people in my university. But during the postdoctoral training in Brazil, before the application, I contacted Professor Rocha, and I contacted him for the possibility of hosting me in his lab. And he said that there are ethical restrictions with respect to the use of rodents, that if I'm happy to work with flies, he will be happy to host me. So I applied, and I was privileged to get the award, and I was in his lab from 2013 to 2014. And I left his lab in Brazil with 2 vials of fly. And from 2014 and 2017, we started from my office. I turned my office to a mini fly lab, and then we started. And then I remember when DrosAfrica team visited, and we were able to get different opportunity to meet with different people within the university. And then the HOD at the time, Professor Olorunshogo, I was able to be given a bigger lab, which is now what we are using currently as our Drosophila laboratory. And this outreach strain brought from Brazil has been distributed to about 15 institutions in Nigeria. It seems to be very, very good because since that time we have not lost these flies. To the question that somebody has the other time, I think the Irish strain is good. It's able to survive since 2014 to 2020. And even 3 African countries have received the flies. So why do we use Drosophila in toxicology? I will only highlight the aspects that relate mainly to toxicology. For example, if I'm to compare Drosophila with rodent that I was using before I started working with Drosophila, you realize that Drosophila has short generation time, high fecundity, and short life expectancy. Another very interesting thing about the fly is the fact that the cost of maintenance is affordable and few ethical concerns, and then it also complies with ECFAM. If you're looking for any model that you can use to replace baby rodent, you can give Drosophila a try. You can start working with Drosophila and leave it. You will definitely fall in love with the fly. The genome of Drosophila is well characterized. For example, we have about 15,500 genes, and for chromosome, if you compare that with that of human that have 23 chromosomes, we have 22,000 genes. There is virtually a match for every human organ in Drosophila. For example, the small intestine painted in red, Drosophila has intestine, and also Drosophila has mammalian-like intestinal system. Mammalian intestinal system, as well as fat body resembling adipose. So it's the fat body that actually carries out the function of the liver. There are different stages, the larva, the pupa, and the adult. The adult stages. There is no stage of the fly that cannot be used in toxicology. For example, the egg can be used to study developmental toxicology. So there is no stage of the fly that is not useful. Drosophila has a very simple, simplified brain, but it's truly complex. If you look at this particular slide, for example, you will discover that the center for learning, for movement, for smell, present in human are also present in Drosophila. It can be through oral injection. It can also be through inhalation of the particular toxicant. But in our laboratory, we focus mainly on the oral route of administration. I like this particular system that was actually developed by Filosevic in 2018. They used this particular drug delivery system to volatilize cocaine by dissolving it in ethanol and putting it in this chamber. And when you exert pressure from this end, the vapor leaves, you remove the clamp, and then this is the activity monitoring system that they devise. So if you are interested in working with this particular area of study by exposing them to fumes, this can easily be improvised. This is a bit complex as indicated in this particular place, the inhalation chamber. We do not have inhalation chamber, so we have never carried out inhalation study. And Amos in 2017 exposed flies to different chemicals, toluene and formaldehyde. They carried out survival tests and macroarray studies to be able to look at the effect on different genes. Is able to carry out several genes at the same time, and then they look at the behavior after exposing the flies to this. This is a mini review that we wrote in 2013 on Drosophila melanogaster as a promising model in toxicology that you may find interesting, especially if you are a beginner in the field. My area of research, as well as my team, actually around these particular two areas. We are interested in understanding the mechanism of toxicity of reproductive toxicants as well as neurotoxicants. And when the mechanism of toxicity is understood, it will be easy to design concepts for prevention and possible therapy. And there are several chemicals in the environment that we have worked with that actually induce toxicity either in the brain or in the reproductive system. With respect to the reproductive system, I particularly take interest in the ones that can affect the female reproductive system. So some of them are listed here: 4-vinylcyclazine and its metabolites, MPTP, rotenone, copper, aluminum, mercury, bisphenol A, and trichloroethylene. The brief methodology of exposure to the fly in our lab is to just expose the fly to the toxicant that is already mixed with the diet at different doses, and then we homogenize depending on the particular assay. That will determine it, because you need to maintain the activity of the enzyme at appropriate speed and temperature depending on what is of interest to you. And then you go ahead to use that sample as any other sample in the laboratory for evaluation of your markers of interest. Depending on your research question, the research question will determine the endpoint markers to use. If your interest has to do with viable cells, you are interested in adaptive responses in viable cells, these are some of the assays that can be carried out. For example, you can decide to look at gene activation or antioxidant enzyme expression. But if the question has to do with cell death, then you can look to look at any of these markers like the 3 activation. With the fly, we didn't just start flying at the time. We started very little. It took us between 2014 and 2014 we started. Our first publication came out in 2017. The major focus was not just was not to publish, but to be able to. I carried out in Brazil, Professor Richard's lab. And then we have survival and longevity assay, behavioral, oxidative stress, antioxidant markers, inflammatory markers, Western blotting, immunofluorescence. When I visited Professor Elan Davis' lab in Oxford, I did for SMC study. So anytime we intend to, not sure of any lab with confocal microscope around. In order for us to ensure that we keep track with the toxicology, we ensure that any of the study that we want to carry out falls among in any of these categories. For example, mechanism of toxicity, safety of chemical products. So in the next few slides, I will be talking about each of these, and I will be telling you how Drosophila has different aspects of this. I will also focus on some of the papers that we have published in the laboratory. As a result of the high number of chemicals available worldwide, there is need to develop sensitive and rapid assays for the screening of toxicants using relevant models. And this is particularly crucial because it's necessary, for example, if a particular product or maybe water is contaminated and you really want to know the mechanism of toxicity, Drosophila seems to be very, very good to be able to do this. So the question now is Drosophila appropriate as a model to investigate the mechanism of toxicity of chemicals? This paper that was published in 2009 by Singh et al., they look at 3 chemicals that usually coexist in the environment, and they are structurally related: benzene, toluene, as well as xylene. And then they use Drosophila as a model to establish the mechanism of toxicity of these 3 compounds. And the finding was that they all induce reactive oxygen species overproduction of reactive oxygen species, which actually eventually led to oxidative stress. Let me mention at this stage that there are several compounds that can truly induce oxidative stress in different ways. So when we say a particular toxicant induces oxidative stress, or maybe 5 compounds induce oxidative stress, it doesn't mean that the mechanism of the toxicity is the same. For example, we have some that can just affect the total tire component of the system. And there are some that can just target some of the same mechanism of toxicity, even though they are all categorized under the same name with respect to oxidative stress. So this is the compound that I work with in Brazil during my postdoc with Professor two metabolites. This 4-vinylcyclohexane can get into the system through occupational exposure. It can also get into the system through consumer exposure. For example, passive emission from carpet result. So if somebody has a new carpet, it's good to leave the windows open for ventilation to take place. People who also work in companies where they manufacture shoe soles may also get exposed to this 4-vinylcyclohexane. It's metabolized by CYP2B to VCD. But in the ovary, VCH can be metabolized directly to VCD. The first paper that we published was published in Periodic Abolaji and Medicine, and the finding was that 4-vinylcyclohexane, the parent compound, cause accumulation of and nitrogen species, and also it also led to the misregulation of, and then we also look at the metabolite after metabolism, and what we did was to expose the metabolite separately to the fly. And one of the key mechanisms of toxicity that we found out was the fact that this metabolite inhibited delta-aminolevulinate dehydratase activity, which is a key enzyme in heme synthesis. And this particular enzyme has zinc at the active site and 3 thiol groups in close proximity, and the VCD now form adults with this biology. If it is used to treat the fly, the negative geotactic acid affects the climbing behavior of the fly, as a fly that is diseased or exposed to the effect on the behavior of the fly. So the next thing is to look at how DrosAfrica can be used to evaluate the safety of chemical products and drugs. By way of a case study, there was something that happened at the time. Permit me to quote some of the names, just to mention that a businessman bought products worth $1,000 US and exported this product from Africa to Europe. After analysis on arrival, they found out that they contain high amounts of poisonous levels of some of the components. And then the man sued the company, and in defense, the company said the products were for local consumption. And the court blamed the regulatory agency in that particular country. With respect to this is the fact that regulatory agencies in African countries should strengthen certain policies, certain regulations, in order to compare manufacturers of consumables to correct inadvertent or intentional errors. This is very, very important. These are 2 herbal products that are widely sold in Nigeria. And we carried out safety evaluation, not for any 14 year, the finding with, and we found that there was no adverse effects were not affected in comparison with the control. However, the glucose level was reduced. The habit of the drug is actually to treat diabetes. The habit drug. So for somebody who is not diabetic that is taking the Ruzu, it may reduce the level of glucose. So caution should be taken while taking the ababita. And then DrosAfrica also as a model to assess safety of food and water. We also carried out some study with respect to this. And the first that we did was to look at monosodium glutamate, which is a signal that is commonly used in Africa. And we found that the flies a period of time at the reactive oxygen and nitrogen species, as well as catalyst activity, GST, and total thiol level increase through the process of adaptive response. However, by exposing the fly to different concentration of monosodium glutamate for a long time, it actually resulted in decrease in the lifespan of the flies. Another study that we carried out in collaboration with a colleague in the department was to look at the role of apigeny that is widely used, that is present in different plants, to look at if it's possible for this to actually be used to ameliorate D-galactose in this toxicity. Because if you look at the study that was just presented, you will look at the side effects of carbohydrates in the system. So we use D-galactose as a particular concentration to induce toxicity in the fly, and that resulted in decreased lifespan, oxidative stress, as well as apoptosis. But by exposing another set of fly only to apigenyl alone, it increased longevity, antioxidant status, and also apoptosis was reduced. The combination showed that the apigenyl actually ameliorated the toxicity that was observed as a result of D-galactose. The next aspect that I'll be talking about has to do with the possibility of ameliorating the toxic impact of different drugs and chemicals using phytochemicals and plant-based products. We prefer to use phytochemicals in our lab because the structures are known and the phytochemicals are present in different plants that are abundant in Africa. And then these are the different toxicants that we have looked at, reproductive toxicants, as well as neurotoxicants. The first that I have here is the exposure of light to trichloroethylene, and trichloroethylene actually disruption of redox balance. However, when co-administered with aspartame of trichloroethylene, aspartame is found in food different. Bisphenol A causes toxicity in the fly by inducing oxidative damage, but when co-administered with luteolin, the toxic effect was actually alleviated. The next on my slide here is aluminum chloride exposed to Drosophila induced oxidative stress, and then curcumin prevented reduction in oxygen emergence that was seen in the group of rats exposed to aluminum chloride. So curcumin prevented aluminum chloride mediated oxidative stress and behavioral deficit. This slide has to do with the rotenone exposing flies to rotenone. Rotenone is one of the chemicals that can be used to induce Parkinson's disease in the laboratory. And colaviron is actually a bifluorone complex from Garcinia cola that was extracted from Garcinia cola. And this prevented the behavioral deficit as well as the inhibition of antioxidant enzymes and redox imbalance observed in the fly exposed only to rotenone. This particular study was carried out in the laboratory of Professor Rocha by Sudati et al., in which Valeriana officinalis attenuated rotenone-induced toxicity in Drosophila melanogaster. So you will agree with me that Drosophila is a good model to study the mechanism of toxicity of compound. MPTP is a neurotoxin and it induces the symptoms of Parkinson's disease via destruction of dopaminergic neurons. And what happens is that the electron transport chain is blocked, and as a result of that, it's also able to cross the blood-brain barrier and then damage the neurons, especially the dopaminergic neurons. And we found that MPTP reduced the survival of the fly after 7 days of exposure and also caused oxidative damage. But when co-administered with grapes with resveratrol is found in grapes and is also found in several other foods. Resveratrol actually increased the lifespan of the fly and also the rescued MPTP-induced Parkinson's disease symptoms in Drosophila. And this was funded by ISN, and we use this project as preliminary data to apply for IGEM grant. And we are currently working on this project to determine the molecular target of resveratrol in genetic chemical-induced Parkinson's disease models. And we are using different genetically modified fly, and also we use different techniques to do that. We are currently working on this. It is true that there is strong correlation by way of conclusion between the fly and mammals. However, we should take note of the fact that metabolic differences may occur in which some drugs or toxicants may be toxic in human or toxic in fly and not being toxic in the other. Despite this limitation, the fly is a good model for toxicology and common pathways or target of a given drug and class of toxic agent. The material that I highlight here under material for reading is just a link to all the Drosophila papers in PubMed that were published. This book is also good for anyone that is actually interested in working with Drosophila. And the fly-based resource is also there. I'm sure this will also be talked about in the course of the conference. I must acknowledge several organizations that have supported our work at different times. I will also have to appreciate several people, that the time will not permit me to mention them one after the other. I must acknowledge all the students from 2014 that have been supervised with the work. I see them as my team. We work together to ensure that we continue with the fly study. And also the current postgraduate students are also acknowledged. I want to thank you for your attention in the course of the presentation and before the presentation. I want to appreciate DrosAfrica for the opportunity of this presentation. I must also thank Professor Ruskegan that I met during the Drosophila workshop in Ibadan. So DrosAfrica has also made it possible for us to do a lot of things, and the network has really been tremendous. I thank you all.
Excellent. So let me just say, so thank you, Amos. That was amazing. And let me say, there's a lot.
Thank you.
There's a lot of, you and I will get a chance to talk some more, but there's a lot of comments here, which I'm just going to echo, which is your efforts in African toxicology are just amazing. That is impressive work. As you say, you had a few network issues, but actually your slides were dead on, so that was not a problem for me. So I appreciate it. It was the first time I really got a chance to see your work all the way out from you. Really, really beautiful work. So I appreciate it. Thanks. And you can read the comments of the people on the side, but let me ask you some science questions. And I'm going to take them in order. And it's my role to keep the discussion short, so we'll move on. So I'm going to be the bad guy on that as well. So from Marianne Adeniki Salaudin, does the fly have an equivalent of the human skin? And if so, basically the question is, how do you evaluate the toxic effect of a product that is used topically on human skins? How do you evaluate that in flies? The question is, so do flies have skin? And basically, how do you use flies for things that are toxic that we test on people's skin?
We have not carried out that study in fly. When we started with simple things. You said if the fly can be used to
things that you put on your skin. Can you use flies to test for toxicity?
We have not done that, but it can be done. We have not done that. We have not done that in the lab. We started with simple things that can easily be carried out, and then we progress gradually.
Got it. Yeah, I think that is a challenge, but because they have a cuticle. But yeah, I agree. It can definitely be done. So I'm going to probably only ask you one more question because we're going to need to move forward. And that is actually multiple people, including Mariam Deniki, Celadine and Audubon, Bismarck, Dekura. Basically, they're asking, how did you choose monosodium glutamate as your toxin of choice? And also it's more commonly found in food than in drinking water. So can you look more at monosodium glutamate and its effects specifically in food rather than just water?
Come again, please.
Monosodium glutamate?
Okay, it's just out of curiosity because people actually use the concentrated, the pure monosodium glutamate in washing, doing different things, and also it's commonly consumed. And then, out of curiosity, we wanted to see the effect on longevity, and we exposed the fly on a short-term basis and a long-term basis. And the short-term basis resulted in adaptive response, but long-term basis actually reduced the lifespan of the fly. It's out of curiosity to be able to understand what happens. Can the fly be used as a model to assess safety of food? So we decided to look at the compound. And it's also possible to expose the fly to any diet containing this monosodium glutamate. The only thing is that you may not know the concentration that the manufacturer used, except you first analyze to determine the level.
Got it. Cool. I'm going to allow one more question for Ismail, because I think it's a really good question. Adeni, so I'm going to pull you a little over time. He says, thanks for the presentation. Is there any neurobehavioral equipment to assess locomotion disorder associated with Parkinson's in Drosophila? So this, of course, is a big one for toxicology.
Oh, yeah. Thank you for the question. It's a very good question. The trikinetic actually tells locomotor activity monitor device. We just ordered for this. It has not arrived. That you can use to do a lot of behavioral studies. And also, it's so interesting with respect to the fact that you just put a fly out of the group into a vial, a small vial, and then you connect it with a monitor. It's a very good way of assessing neurobehavioral effects of drugs. So, kinetics, you can just go online and then you can contact them.
Nice. So you're going to be studying this. Very nice. All right. Well, we have to move on. Thanks, Amos. Much appreciated. And don't forget, there's been a number of requests for posting the papers that you put up. So if you can put those in Slack, that would be great. Or if you just, I guess if you post them in the chat, people can probably grab it as well. Okay?
Okay.
All right. Thank you very much.
Thank you very much.